continue on with our next presenting company, I'd like to welcome up Immuneering Corporation. Presenting for Immuneering Corporation is Harold Breakwood, Chief Business Officer.
Hi everyone, and thanks for that introduction. Thank you for your time today, and I look forward to sharing with you some important information in the advancement of pancreatic and other cancers. Immuneering there we go. So I'll share with you overall survival data, tolerability data, our plans for pushing forward, and the significant opportunity for patients, investors, and partners with Immuneering's program. Kind of just getting used to the clicker here. It's a little slow to respond as I push forward. There we go. So this is a Kaplan-Meier curve with atebimetinib plus modified gemnab paclitaxel. This is in first-line metastatic pancreatic cancer. This is a striking, extraordinary curve where we have recently shown 9-month data in this patient population with an 86% overall survival, 86%. We had previously shown back in June a 94% overall survival. This is at the 320 milligram dose of atebimetinib in 34 patients. So really kind of an extraordinary result. Let me put that into context. So if you can see the orange line below the curve that I just showed, that's from the MAPK study, which was published in 2013 in the New England Journal of Medicine. And this is an indirect treatment comparison, so with all of the caveats that come along with that. But at the 6-month mark, there was a 27% gap between the two curves. And at the 9-month mark, that gap has actually widened to 39%. So significant gap between the two curves. And I'll go into some of the details in terms of the patient demographics and things like that that you'll see here. But we're very excited about these results and the difference in the potential improved outcomes for patients. And on the basis of this, in the recent weeks, we've raised $225 million from various investors. It's not just the MAPK study, but here I'm showing basically the 4 standard of care curves. So this is FOLFIRINOX, NALIRIFOX, gemnab, and a modified gemnab. So it kind of, all 4 of those standard of care curves basically cluster together. And there's a huge gap of our product versus those other 4. So it kind of doesn't matter which of those 4 regimens one picks. There is a significant improvement versus each of those standard of care. Similarly, with progression-free survival, and I just have to be careful not to double-click this. So this is, again, in the same 34 patients. This is the PFS curve. And you can see here at 9 months, it was 53%. And we actually have reached a median PFS. We had not yet reached a median OS in the prior curves that I showed. The median PFS is 9.6 months, which if you can see over here, it's significantly higher than the other kind of landmark studies. And again, if you do an indirect treatment comparison with the MAPK study, significant gap, both at the 6-month point and at the 9-month point. And similarly, the other 4 studies kind of cluster together. There's a significant gap on PFS. So let me show you the other efficacy parameters. So this is the waterfall plot, and there's a few things to kind of point out on this very busy slide. First of all, there's not much of a left-hand side to the waterfall plot here. So almost all of the patients had tumors regressing. So that's the first thing to point out. The second thing is the ORR is 39%. And I'm sorry I don't have a clicker. I know these are busy slides, but 39% for atebimetinib in comparison to the MAPK study, which was only 23%. So we've nearly doubled the ORR. And the disease control rate is significantly higher as well at 81% versus 48%. A couple of other things to point out on this. Um, the diversity of patient mutations, so along the horizontal axis, this is an all-comer study. We didn't select patients for specific tumor mutations. So there's a whole variety of G12D, G12V, G12S patients. It was essentially all comers regardless of mutation status. And I guess the last thing that I would point out is there are a number of patients who continue on study. If you can see the little arrows on that slide, that denotes the patients who continue on therapy. So since we last disclosed results in June, we've had a number of patients who had unconfirmed responses convert to confirmed responses. And we're hopeful that a number of the patients who have stable disease now will convert to partial responders as well. So kind of going on to the spider plot now. So kind of the takeaway from this spider plot is somewhat the slow and steady nature of what atebimetinib is doing here. You kind of see a gradual reduction in the target lesions. And I'll show you a couple of more things that kind of emphasize that kind of slow and steady efficacy of the drug. But first, one of the towering strengths of this program is the safety and the tolerability. We specifically designed atebimetinib, which fundamentally targets MEK, to pulse the MAPK pathway, which is very, very different than other products in this area. And we did it for a very purposeful reason. MEK is a great target, but it's been really limited in its clinical utility over the years because of the toxicities, and it's kind of been relegated to BRAF melanoma and other indications. So we designed the molecule to have certain properties, including a high Cmax, a very short half-life, and a near zero drug trough every single day. And it's paying off. You can, hopefully you can see on the far right-hand part of this slide, these are the AEs, and it's a 10% cutoff, which is kind of the typical cutoff, and it's grade 3 or above AEs. And many of those numbers in that blue column are 0, which is kind of extraordinary for taking a MEK inhibitor and combining it with chemotherapy, especially in comparison to the other 3 standard regimens that we show there from those publications. I mean, a couple of things pop out. We get a lot of questions about kind of the neutropenia and the anemia, and those are really kind of classically associated with the chemotherapy and really the chronicity of the chemotherapy. So the good news is because atebimetinib is active and so well tolerated, we're finding patients stay on the regimens longer, and I'll show you kind of an extraordinary example of that in just a second. So usually at this point in the presentation we get asked, well, is there some kind of special benefit in the patient demographics that you studied? Did you kind of cherry pick the population or something like that? And the answer is no. In fact, we had an older population than many of the other studies. And older patients are prone to comorbidities and other issues. So the median age of our population was 69 compared to kind of lower 60s of the other 3 pivotal studies. And 68% of the patients were 65 or older, which is, again, a significantly higher percentage compared to the other studies. One of the other questions that we get asked is kind of like, what was the location of the MET? Because there's been some recent studies where certain drugs have worked better in with lung mets, which tend to be more responsive. And so we've analyzed our results and specifically showed that there's basically no difference in the results between lung met, liver met, and peritoneal mets. So it's a consistent result across the different areas. So this is where we're going next. We're super excited about being able to do a pivotal registrational trial in first-line pancreatic cancer. We all know how devastating pancreatic cancer is, and we have family and friends that have been subject to this kind of horrible tumor. It's got the lowest 5-year survival rate of any of the major tumors. There really hasn't been any new therapies in decades in this. And so we plan to initiate this study this year and to dose the first patient by mid of next year. And so this trial, and again, it's in first-line pancreatic cancer, where we really are the leaders in first-line pancreatic cancer. We have a dose. We've demonstrated now survival in first-line. We've demonstrated the ability to combine with chemotherapy, and we're excited to basically bring that to a registrational trial, where we're going to continue doing what we've been doing, which is the experimental top blue box, atebimetinib plus gemnab, and compare that with the standard of care gemnab. And the difference is in the dosing frequency. We dose 1 week on, 1 week off, versus 3 weeks on, 1 week off. And OS will be the primary endpoint, which is what everyone wants. Patients want it, providers want it, regulators want it, HTAs want it. And then the usual secondary endpoints with PFS, ORR, disease control rate, and quality of life. As I said, we're going to initiate this by the end of this year. We expect a top-line readout by mid-2028. So just real quickly, because usually at this point people ask, well, what have you done differently? How is this drug different than what else is out there? In kind of a nutshell, it's all about this deep cyclic inhibition approach. On the left-hand side, there's basically, kind of with the prior generation, products, it's all about sustaining occupancy of the target and inhibition. And that basically creates resistance. And so there may be tumor shrinkage initially, but then it's not sustainable. Whereas what we're doing with deep cyclic inhibition is basically pulsing the pathway at the target rapidly, and basically preventing cancer from adapting, which creates a more durable response and a better tolerated response. And that was from a publication by Gatenby et al. At Moffitt. So there's some additional evidence in this area. So this is the patient that I mentioned. So this is kind of an extraordinary case study that we shared at ESMO a year ago and in other places. So this is a third-line patient. So this patient had failed FOLFIRINOX. They had failed a gemcitabine-based regimen. And this is monotherapy, so there's no chemotherapy on board here. So this is just atebimetinib by itself. This is an older 70-year-old man who actually was given the 240 milligram dose of atebimetinib, so actually the lower dose which we use for our lead-in. And he's just had a very, very good experience and a good result with this in terms of quality of life. He's actually gained weight, which is not a fluke. There's a mechanistic basic reason for the weight gain. Significant reduction in his CA19 line, 19-9 levels. And this was a KRAS G12D patient. He had a complete resolution of a bone lesion. And you can see that from the kind of the single patient spider plot, he didn't become an official RECIST responder until 14 months, which is kind of consistent with the way the drug is working. And anecdotally, when he was told that he was a responder by his provider, he said, well, what do you mean? I've been responding to the drug. I've had a very good experience. He was on the drug for over a year. And this is somebody who probably would have only expected about 4 or 5 months as a third-line patient to survive. And now the patient is out 17 months and counting. Very positive experience there. I'm going to skip this in the interest of time, but this basically in a nutshell shows how we're shutting down the MAPK pathway on the left, as opposed to the single mutation products which have additional mutations. So pancreatic cancer is just the beginning. We're also excited to get going on a couple of lung cancer studies. We have an arrangement with Regeneron to combine with Libtayo, and we'll study in, wrap up? Okay. What's that? Oh, that restarted. Okay, I'm getting the proverbial hook. Okay, I will just end with that picture, which was a picture of our lab site in San Diego. The molecule is discovered and developed by us. Sorry for going over, and thanks very much.